Hmm. Okay, let's get started. All right. So, good morning and welcome to the Early Career Scientist Symposium on the topic of phenotypic plasticity. I am so excited to see so many faces here, especially at 8:45 on a Saturday morning. And really thrilled about the program we have ready for you today. We have some really fabulous early career speakers who've flown in from all over the country to share their stories with you. To get us started, I wanted to just sort of spend a minute with these words from Mary Jane West Eberhardt, which I'll go ahead and read to you. um, the environment is not only the agent of selection, in the sense of being the arena where different phenotypes achieve different degrees of survival and reproductive success. It is also an agent of development, which importantly influences the range of phenotypes that will be produced by a given genotype. So I love this quotation by Mary Jane West Eberhardt because I think it beautifully disentangles the distinct ways that environments might shape phenotypes, both as kind of the container in which organisms do all their developing and expressing of a genetic program, and also as the filter that chooses which of those organisms get to pass genes on to the next generation. So today we're going to hear about environments as agents of development over and over again from Laura Stein, from Ben Parker, from Holly Muller, and Claire Ritchoff. But we'll also hear about the environment as an agent of selection from Yuheng Huang, Cameron Galambor, and Juana Carha. But today we're also actually going to add another dimension in that we'll think about how plasticity can feed back to shape the environments that organisms are doing their development in, sort of via the niche construction that Daniel Schwab will speak about, and also the trait-mediated effects on ecological processes that Karen Burkhardt is sharing. So I'm thrilled to hear what they all have to say, and want to start by just highlighting these two people, Dr. Nancy William Walls and David Bay, who are responsible for us being here today. Nancy William Walls was a microbiologist trained at University of Michigan. She went on to be one of the first female faculty members at the Georgia Institute of Technology. And she helped to establish what was called the Young Investigators Symposium about 13 years ago. Then more recently, support for the symposium comes from the David Bay Photography Fund. David Bay was a much beloved photographer at large for 34 years for the department. And a fund established in his name is helping to fund us today. So I believe our chair, Dear Midfogel, will have a few more words about them later on in the afternoon, but I just wanted to highlight their contributions to get us started. So today, here's our itinerary. We'll have 3 talks in the morning, followed by a coffee break, then 2 more talks. At noon, we'll actually invite our morning speakers back up to hold a discussion so that we can invite people to ask questions you don't get time to have answered during that panel, or we can have sort of a broader, more synthetic discussion. We'll see what comes up. And then in the afternoon, we have 3 great talks, followed by a coffee break, then our 2 last speakers, including our keynote address. We'll have a final panel discussion and then move on to the Natural History Museum for more conversation over drinks and snacks tonight. Speakers for the afternoon, please make sure that you come up and get your talks loaded during the lunch and poster session. We have decided we're going to give you a 5-minute warning and then no 1-minute warnings, because I hear that's what you've requested. But if you run out of time, we will stand up. Sonal, Mary, and I think, are responsible for that, and we are very fierce, so please don't run over time. If the speaker leaves time for questions right after their talk, we'll circulate some microphones so we make sure we capture your questions. We are recording today, so if that changes anything, just I want to let you know. If we don't have time for questions following a talk, there will be an opportunity to ask those questions at the panel discussion. So other details, we have a ladies' and men's restroom on this floor just around the corner here. There's a gender-inclusive restroom on the 3rd floor. The Natural History Museum is just around the corner. There are maps on the back of the programs out on the registration table if you need help getting there. Um, let's see, what else do I need to tell you? Oh, um, finally, at the end of the day, if we run out of time and you still want to keep talking, I think we're going to plan and gather after the reception at MASH on 2011 East Washington Street, where you can buy your own drinks and keep talking. And then before we get started, I just want to let you know that we're actually trying a new experiment this year. So we have a coffee break assignment for you. So out in the hall, there's a big sheet of paper and some Post-it notes and markers. If you have questions that come up during the talk or sort of more general things that you'd like to discuss on the panel, we invite you to write those questions on a Post-it note or on the piece of paper, and we'll make an effort to sort of collect those ideas and sort of inquiries and bring them into the panel discussion. So this is one way to get more voices in the room in conversation. So thank you for being here. I think Mary is now going to introduce our first speaker.
Okay, so our first speaker of the day is going to be Maggie Wagner. Maggie is an NSF Plant Genome Postdoctoral Fellow working at North Carolina State University. She studies how complex plant traits evolve in heterogeneous environments with a special focus on plant-associated microbial communities. Maggie got her bachelor's degree in plant science here at the University of Michigan, so she obviously got off to a great start. She earned her PhD at Duke University under the mentorship of Thomas Mitchell-Olds, and today she'll be sharing part of her dissertation work where she investigated the role of genotype by environment interactions in the evolution of defensive chemistry in a wild perennial plant. So thank you, Maggie.
There we go. Okay. All right, thanks, Mary, for that introduction. It's true that I now mostly work on plant microbiome variation, and specifically I'm interested in how host genotype interacts with the environment to shape plant-associated microbial communities. I'm not going to be talking about any of that today, but I love to talk about it, so if you want to catch me later to chat about that, I would be delighted. What I'm going to be talking about today is part of one chapter of my dissertation that I did at Duke in Tom Mitchell-Olds' lab. Anyway, it's really cool to be back at my alma mater, so thank you to the planning committee for inviting me and for all your hard work putting together this great event. I'm really looking forward to the day. And before I get started, I want to acknowledge the contributions of my PhD advisor, Tom Mitchell-Olds. His guidance was critical in helping this project come together, as well as my former colleagues at the Mitchell-Olds lab. We all went out to Idaho every summer and did our field season together, and there's no way I could have done this scale of field experiment all by myself. And so their help and collaboration was really critical. Okay. So the natural landscapes where organisms evolve can be really complex. So for example, in this photo of the Salmon River Valley in central Idaho, you can see alpine areas, you can see dense softwood forests, sunny high elevation meadows, and 2,500 feet below is the Salmon River itself, which is relatively warm and wet compared to up in the mountains. And there's also a lot of invisible environmental variation in this landscape, things like soil chemistry, differences in insect communities among habitats, differences in microbial communities. And plants are sensitive to all of this. And so how does all of this environmental variation affect the evolution of the plants that are native to this area? Well, environmental variation plays two key roles in the evolution of organisms. First, it determines the phenotype expressed by a given genotype, and this is plasticity. This is the theme of this symposium. But it also determines the adaptive value of that phenotype. That's natural selection, and I think most people intuitively understand that different types of environments select for different sorts of adaptations. What's less clear is what happens when these two roles intersect. How does plasticity affect the course of evolution? Now, a lot of theoretical work and empirical work has explored this concept, but there's still not enough data from natural habitats that can be used to test these predictions. Now, I'm going to back up and start by quickly talking about one way we can detect plasticity, just to give some background for my talk. So when we're thinking about a continuous or semi-continuous trait, such as the number of spines or the concentration of a defensive chemical or an organism's height or something like that, how do we tell what causes all this variation contained within this bell curve? Well, we have to compare the means of different groups or categories. So for example, these red and blue groups could represent two different genotypes. So in this example, on average, the blue genotype invests more in defense than the red genotype, which indicates this trait's partially genetically controlled. Or these red and blue groups could represent two different environments. And assuming the plants in both environments are genetically identical, this means that the trait shows phenotypic plasticity between these two environments. Now, when there are multiple potential sources of variation that all interact with each other, it's harder to visualize on a graph like this, but luckily we have well-established statistical tools that allow us to tease them apart. So with a well-designed experiment, we can use analysis of variance to partition the trait variance among multiple interacting sources. So what this equation says is just that the total variance in the phenotype is the sum of the variance contributed by the organism's genotype, plus the variance contributed by the environment, plus the variance contributed by the genotype by environment interaction. And both these E and G by E terms describe different aspects of plasticity. So for example, we might observe only an effect of genotype with no plasticity. So if you imagine an experiment with two genotypes measured in two different environments, the red and black genotypes here are the mean trait values are shown for a red and black genotype in both habitats. And you can see in this case, there's a consistent difference between the two genotypes, but the environment makes no difference. You could have the opposite, where there's no difference between the genotypes, but in habitat 2, trait values are consistently higher than in habitat 1. And this is an example of plasticity, or just an environmental effect. You can also have both genotype and environment effects. So there's still a consistent genotype difference in both habitats, and habitat 2 is different on average than habitat 1. And finally, you can have a genotype by environment interaction. And this is a way of saying that the genotype difference depends on the environment. Another way of saying this is that plasticity depends on the genotype. So for example, the red genotype has the same trait value in both habitats, but the black genotype has a drastic change in phenotype when moving between environments. Okay, so that's a little background, refresher on quantitative genetics to help you follow my talk a little better. So what happens when plasticity and selection intersect? Well, you can imagine that there could be two genotypes that are well adapted to their habitat. This blue star in this graph represents a phenotypic optimum. Now imagine that the population encounters a new habitat. For example, maybe they move from their original habitat to a new place with no predators. And this could either be colonizing a new area, or it could be a drastic shift to their actual environment. For example, maybe climate change is driving out a natural enemy population or something like that. Now, defense traits are often costly, and organisms have to balance that cost with the benefit of resistance. And so if the new environment has no predators, the optimum investment in defense might change. And how will plasticity affect how the organisms do in this new environment? Well, there's two different patterns that could happen. The red genotype here manifests what is called adaptive plasticity, which means its phenotype moves closer to the new phenotypic optimum. In contrast, the black genotype moves farther away from the new optimum, and that's called non-adaptive plasticity. Now, those names are slightly misleading because actually either of these patterns can possibly facilitate local adaptation. So adaptive plasticity causes a short-term increase in relative fitness. It allows the population to persist when it otherwise might have gone extinct in a challenging new habitat, which might allow it to persist long enough to adapt genetically to the new habitat. But non-adaptive plasticity, although it decreases relative fitness in the short term, it intensifies natural selection, which could cause rapid evolution and rapid genetic adaptation to the new environment. And which of those happens is a big issue that is actually beyond the scope of my talk. I think our keynote speaker, Cameron Gallenbohr, has done some really great work in this, and maybe we'll get to hear about it later, I'm not sure. But anyway, what I'm going to talk about today is just what are the relative frequencies of these two patterns of plasticity across a complex landscape. And there's surprisingly little data on how often plasticity is adaptive or non-adaptive in natural habitats, especially we especially lack data for physiological traits like chemical defenses, probably just because they're harder to measure, really. So this is the general approach that I'll be taking. The first step is to determine the direction of plasticity in each environment. And the second step is to determine the direction of selection in each environment. And once we know that, we can then ask, okay, how well or how often does the direction of plasticity match the direction of selection? So to answer these questions, I did some experiments with this cute little wildflower called Boechera stricta. It's native to a diversity of high elevation habitats in western North America, from Arizona up through the Rocky Mountains. But I did my experiments in central Idaho, which is right in the center of genetic diversity in the native range. It has two ecologically divergent subspecies, which are named East and West. We like Boechera because it's experimentally tractable. It's a relative of Arabidopsis, actually, a wild relative of Arabidopsis. And it's not quite as experimentally tractable, but it's pretty easy to work with, nice and small, and you can grow lots of them relatively quickly. But most importantly, it's naturally inbred, which means that we have access to a big diversity of naturally occurring inbred lines. And the reason this is important is because to study plasticity, it's important to hold genotype constant, or at least to control for genetic variation in order to tease apart which of the phenotypic variation is coming from the environment versus genes. And since these are naturally inbred, we can essentially treat sibling plants as genetic clones of each other, which is very handy. So the trait I'll be focusing on are glucosinolates. And glucosinolates are a family of defensive chemicals, the main family of defensive chemicals in the Brassicaceae. These are the same chemicals that make broccoli and cabbage and other brassicaceous foods taste the way they do. And they're important defenses against herbivores and pathogens alike. And all parts of the plant contain glucosinolates, but I'll be focusing on the rosette leaves today. Okay, so what I did was I planted many replicates of 25 genotypes of Boechera stricta into 4 common gardens. So on this map of central Idaho, the red and black pins show the locations where these genotypes were originally collected out of their native populations. And the white triangles show the locations of the 4 common gardens. These genotypes were evenly split among the 2 subspecies. The idea here was just to sample a breadth of genetic and environmental diversity to make it as realistic and comprehensive as possible. And I want to draw your attention to the abbreviations in the top right-hand corner. Those just refer to the 4 field sites where I did this experiment. So we planted these 4,000 individuals in the fall, and we came back the next summer to measure them. So this is what the field sites look like during the summer. So 2 of them were high, dry mountain meadows, which are typical habitat of the east subspecies, and the other 2 were lower riparian sites that are more typical western habitat. All of the sites were extremely remote with little to no history of land use, which means that we could observe the plant's behavior in an evolutionarily and ecologically realistic setting with as little human disturbance as possible. And so the plants were in randomized blocks planted directly into the natural vegetation, disturbing the habitat as little as possible. I collected rosette leaves and extracted glucosinolates after bringing them back to the lab at Duke, and then measured those glucosinolates using HPLC. Also in the field at the end of the summer, I measured fecundity, which was just the total amount of fruits produced by each plant, which is a component of evolutionary fitness. So from the glucosinolate data, I can determine the direction of plasticity in each habitat. And by combining the glucosinolate data with the reproductive fitness data, I can actually measure the direction of natural selection in each habitat. And once I have both of those things, we can ask how well do they match. So how do we actually quantify glucosinolate profiles? Well, one thing we have to know is what's the total concentration of glucosinolates, what's the total investment in these defenses. But quantity is not enough because these plants and most brassicaceous plants make several kinds of glucosinolates. And these are the 4 main ones in Boechera stricta leaves. And they're all structurally related except for differences in carbon chain elongation and modification of different side groups. And most relevant for us is they differ in the amino acid precursor used to synthesize them. So the one circled in red, 6-MSOH, is derived from a straight carbon chain amino acid, methionine, whereas the other 3 are derived from branch chain amino acids, valine or isoleucine. And the reason this matters is because these 2 categories of glucosinolate have different biological activity, and so we also need to know the relative amounts of these groups. So I define the trait BC ratio as the total proportion of the glucosinolate profile that consists of branch chain glucosinolates. And we already know from several field experiments in the Mitchell-Olds lab that plants with high BC ratio are more resistant to insect herbivory in many but not all environments. And we also have some evidence that high BC ratio could be costly under certain conditions, especially drought-like conditions. So this is actually a bar plot showing the BC ratio for 1,500 individuals that I measured. And they range from pure methionine-derived glucosinolates on the left to pure branch-chain glucosinolates on the right, with lots of intermediate BC ratio individuals. And finally, there's also lots of diversity within the branch-chain component. And we know that having multiple defensive chemicals can act synergistically to improve insect resistance, so it might be useful to know how diverse these glucosinolate profiles are. And so what I did is I defined the trait branch chain diversity as using the Shannon Diversity Index from Community Ecology and just applied that to the branch chain glucosinolates. So this measure describes not only how many different branch chain compounds there are, but also how evenly they contribute to the branch chain component. So if you only have one type of branch chain glucosinolate, you have 0 branch chain diversity. But adding another branch chain compound increases BC diversity, but only a little bit if one compound still dominates. And it's maximized when multiple compounds are evenly represented. Okay, so all 3 traits plotted together make it pretty clear that there is tons of variation for all of these 3 traits. and also that they're largely independent of each other, which means that we need to assess plasticity of each trait individually. And my experimental design allowed me to separate correlation from causation. So I used ANOVA to partition glucosinolate variance, and I'm highlighting the environmental variance term here because this is the one that's going to tell us about the average plasticity in this species. But the key is that this method allows us to control for genotype differences, since we know the genotype of each individual plant that we measured. So looking at the genetic variance component first, what I've done is plotted the mean value of each trait for each genotype with a 95% confidence interval. And so these are the genotype trait values averaged across all 4 sites. And what this clearly shows is that all 3 glucosinolate traits are under strong genetic control. And now for the plasticity, this time I'm going to plot the mean trait value at each site averaged across all the genotypes. And what you can see here is that there's also strong plasticity in addition to the genotype effects, there's also a strong environmental effect. So for example, BC ratio can change by up to 9 percentage points between habitats, which is quite a lot. And the total glucosinolate concentration at plants at Mahogany Valley produce barely half as much total glucosinolates as plants at the other sites. Glucosinolate profiles were also highly plastic within habitats on a meter scale, but I'm not going to be talking about that or showing that data today, but feel free to ask me about it later. And I'll come back to this key result. This is part of what we'll use to answer our question about the relative frequency of adaptive versus non-adaptive plasticity. And finally, I'll briefly talk about the genotype by site interaction. We're not going to use this result to answer the main question about adaptive versus non-adaptive plasticity, but it's important because it represents genetic variation for plasticity. So for each individual genotype, we can look at how their glucosinolate profiles change when they transition between habitats. So here I'm going to plot the mean trait values for all 4 sites for all 25 genotypes. They're separated by dotted lines on this graph. But we can focus on 2 contrasting genotypes just to illustrate the point. On the left there is a genotype that shows absolutely no change in BC ratio among sites. And the one on the right shows a pretty drastic change in this trait among sites. We can do the same thing for the other 2 traits. And note that genotypes can differ not only in whether or not they're plastic, but also those plastic genotypes can differ in the rank trait values among sites. So the leftmost plastic genotype here produces the most glucosinolates when it's at Jackass Meadow, that black site. And the one on the right produces the most glucosinolates when it's at Silver Creek. And branch chain diversity shows similar patterns. The takeaway here is that there is lots of genetic variation for phenotypic plasticity of glucosinolate profiles. Now, to answer my main question about how relatively common are adaptive versus non-adaptive plasticity, I'm going to be using just the average plasticity across genotypes. But it's important to keep in mind that not all genotypes are capable of that same plastic response. Okay, returning to our broad strategy, we want to determine a direction of plasticity in each site. And in this case, there's no obvious old versus new habitat. Instead, these sites represent the variety of available habitats to be colonized by migrating Boechera stricta populations. And so to determine a direction, we need to define some baseline for comparison. And so I chose to use the experimental grand mean trait values for each of these traits, which I've added to this graph as a gray dotted line. And so just to reiterate, this is a result from the ANOVA showing the average plasticity across all genotypes for each of the traits. And you can see that in several cases, 8 cases, there is a significant deviation from the baseline trait value caused by moving to a particular site. And it's important to acknowledge that these conclusions will be sensitive to the choice of baseline. So if for some reason these habitats were not representative, the conclusions could be misleading, although I don't have any reason to expect that that might be the case. Okay, so we have measured the direction of plasticity. Now we need the direction of selection. Measuring natural selection is a matter of determining a relationship between evolutionary fitness and trait values. This is a method called phenotypic selection analysis. So the original measurement I'm using is the amount of fruit produced by each plant, which is a good predictor of the number of offspring in this species. And so I express that number as fecundity relative to other plants in the same site, and then regressed that measure of evolutionary fitness onto the glucosinolate trait values. And I did this separately for each site. And so what we end up with, for example, at Alder Creek, you can see that the blue line for total glucosinolate concentration has a negative slope. And the way to interpret this is that plants produced more fruits if they made less glucosinolates. And the positive slope for BC ratio, the green line, indicates that plants with more branch chain glucosinolates were more evolutionarily fit. And so I did this for all 3 traits at all 4 sites, and you can see there are some differences there. After correcting for multiple tests, I removed some of those lines had slopes that were statistically indistinguishable from 0. So I would interpret that as no directional natural selection acting on those traits at those sites. But I still ended up with 7 instances of detectable directional selection on glucosinolate traits. Okay, so we have the direction of plasticity in each habitat. We have the direction of selection in each habitat. And now we just have to look at how well these match. And so recalling our results on the direction of plasticity, the up and down arrows determine the direction of plasticity, and so we have that information. Our linear regression of reproductive fitness onto trait values gives us slopes for these lines, which are equivalent to the direction of natural selection on these traits. And we can see that there are 5 instances in which we have both detectable plasticity and detectable directional selection. And as you can see, in all 5 of those cases, the directions of plasticity and selection were the same. What this tells us is that adaptive plasticity is more common than non-adaptive plasticity in this system, at least during this year. And assuming that these sites are representative of the whole species range, it indicates that glucosinolate plasticity is likely to help populations survive in new habitats, at least in the short term. Whether it encourages local adaptation through changes in allele frequencies and genetic evolution is still unknown. Okay, so thank you for listening. If you have any questions that we can't get to today, that's my contact information. Happy to take questions now, though.
What kind of variation do you see in the abundance and diversity of herbivores in these habitats? Because it seems to me that that's part of this equation. If these compounds are designed to resist predators, how does that fit into the picture?
There are definitely, okay, I will say we had a very hard time getting solid data on how the insect communities differed among these sites. but anecdotally, I can say that there were strong differences in just the dominant herbivores in different sites, and the timing of maximum herbivory and things like that. The reason that I chose to use reproductive fitness rather than insect damage to look at this is because we do have some evidence that suggests that insects aren't the whole story. So for one thing, there are also pathogens in this site, and there's also physiological costs to making these compounds that all sort of intertwine to affect selection on these traits. And so to get the evolutionary to get to the bottom of what's actually happening in these plants' evolution, we went straight to their reproductive fitness, which is really what you have to look at when you're talking about allele frequencies changing over time.
I think in your data you have different genotype with different reproductive fitness. And I wonder, for those genotypes that are relatively fitter across all 4 sites, are they more plastic in general, or they are less plastic in general?
Yeah, it's a good question. I did not detect any consistent pattern with that. And I also, it's well beyond the scope of what I showed here, but I did do genotypic selection analysis as well to try to detect selection for or against plasticity, and found nothing. So there was no consistent relationship in terms of short-term fitness with plasticity, yeah.
Great talk, Maggie. So these glucosinolates are also, there's an induction, right? And when the herbivores damage the leaves, does that play any role in sort of the patterns of either I guess I'm just thinking constitutive versus sort of induced defenses, and how that might kind of play a role in this story?
Yeah, that's a great point. Yes, so glucosinolates are both constitutively produced, but also have an inducible change in production. So in this particular experiment, I made sure to sample these leaves very early in the season before peak herbivory to try to minimize that complicating factor. Yeah, the induction of glucosinolates after being attacked is another form of plasticity, which I wasn't able to really look at in this experiment. That and also just even in the absence of attack, temporal changes throughout the growing season in glucosinolate production. And I think I forgot to mention this, but also as the plant develops and goes through different developmental stages, the allocation of and production of glucosinolates between different parts of the plant changes. So yeah, the picture is actually much more complex than I presented it here. I did control for developmental stage in my statistical model. I left it off the slide for simplicity, but yeah, all of that's going on and will definitely complicate the picture.
Okay, is there maybe one last question?
And maybe you provide the information for elevation at those sites, but I missed it. So I just wonder, do glucosinolates also act as UV filters?
Oh,
Because the brassicacea typically are in the northern latitudes. As the season changes and the intensity of light also varies quite a bit. Normally, I understand it's mostly about defense against herbivore attack.
I mean, I'll be perfectly honest with you. I don't know the answer to that question. Yeah, we usually, I guess we're usually pretty focused on the role of glucosinolates in biotic interactions for the most part. Yeah, they affect herbivores, they affect pathogens. There's even been studies showing they affect the rest of the microbiome, non-pathogenic microbiome. But I don't know anything about UV.
That's the sort of conventional wisdom. I mean, normally UV filters are flavonoids, but it's just, as you can see, as plants evolve, particularly at the angiosperm stage, a lot of this interaction happens.
Thanks, that's a good idea.
Okay, so thank you, Maggie.
Okay. Yuheng Huang earned his PhD at the University of Toronto, working with Anil Agrawal, and he is now a postdoctoral fellow at the University of Wisconsin in Madison, working with John Poole. Yuheng's research examines the effects of temporally and spatially varying environments on different aspects of populations, including genetic variation, adaptability, and phenotypic plasticity. He approaches this topic using quantitative genetics, genomic and transcriptomic approaches, using both experimental and natural populations of fruit flies. I look forward to learning more about this work in your talk.
Thanks. The organizing committee invite me to present this work. This is done with my PhD advisor, Anil Agrawal, at Toronto. And this talk, the main questions of this talk is relatively similar to Maggie's talk, but I'm looking at experimental population and in a relatively long time scale. So at the end of this talk, I may mention a related project that I'm doing as a postdoc in Wisconsin. So plasticity is a phenomenon that is widespread, not only widespread across many different species, but it could happen for many different kinds of traits, such as the biomass of the plant that is grown in different chambers.
I'm going to put a mic on you, sir.
Or the pigmentation of the bugs that vary different in temperature. Or the Thanks.
Let's take this off too. Okay. Thank you. No problem.
Thank you.
Okay, there we go.
Or the morphological plasticity when the Daphnia encounter the predators. But to produce different phenotypes for same genotypes in different environments, one essential intermediate steps is changing gene expression. So studying the expression plasticity could provide us important insights on how the genotypes interact with the environments, producing different phenotypes. And second, the sequence technology allows us to measure a large numbers of transcripts in a relatively unbiased way, whereas the traditional phenotypic trait may focus on some traits that we know some prior knowledge of their importance. But just keep in mind, the ecological importance of the transcript trait may be less obvious compared to those traditional phenotypic traits. So in today's talk, I will try to ask two related questions on how the evolution of expression plasticity. First, if a population encounters a novel environment and exhibits some initial plasticity, is there a relationship between those initial plasticity and the subsequent evolution change? For example, the subsequent evolution may reinforce the initial plasticity, for example, by the genetic assimilation, or oppose the initial plasticity. We normally call this counter gradient. And second, in theory, we think that the adaptive plasticity could only evolve when the population experiencing varying environment, the heterogeneous environment. So how can expression plasticity shape adaptively in heterogeneous environment, and whether different forms of heterogeneity matters, such as we know environment could vary through time or over space. So to examine these two questions, I use experimental evolution approach in fruit fly, which is modern organisms, Melanogaster. So here's the history of those populations I'm using. So a large numbers of fruit flies are sampled from the wild and maintained in a benign laboratory environment. We call this grand ancestor. And then a subset of flies are sampled from grand ancestor and maintained in high cadmium diet. We call this ancestral cat. And then a subset of flies was sampled from grand ancestor and maintained in high salt diet. We call this ancestral salt. The main reason of choosing these two chemical stress is just to create a different selective environment, and they are relatively easy to use in the lab. So we cross these two ancestor population and divide the F1 randomly into 4 selection treatment. The first 2 treatments just simulating a constant environment where the population are always experiencing cadmium or salt diet. And this one is simulating a temporal heterogeneity where the diet are alternating between generations. So one generation in salt, one generation in cadmium, and then back to salt and cadmium. And last one, we call this spatial heterogeneity. Basically, population are divided into 2 teams. One is in salt, one is in cadmium. To produce next generation, we mix the individual with equal numbers from the 2 teams and go into the next 2 teams in next generation. And the population size is about 450, and there are 5 replicates population per treatment. And another piece of the information about this population is after they evolve for about 15 generations in this alternative treatment, the survival show a signal of local adaptation. If they are assayed in the cadmium diet, those cadmium constant population do very well relative to the salt population. And the dot is just represent the mean survival per replicate population. And these patterns reverse when they are assayed in the salt diet. For the heterogeneity population, they do pretty well in both diets, suggesting they are relatively well adapted to both diets. So to study the gene expression plasticity, we basically collect newly hatched larvae for each of the population, including the 5 ancestor population. And then we basically expose them to either cadmium diet or the salt diet for 12 hours, and then we sequence the transcriptome from the 2 samples. So you can think about for the same population, just the same set of genotypes that is being put into 2 kinds of diet. So the basically differential expression between these 2 diet samples for the same replicate population could be viewed as a measure of expression plasticity. Okay, so first is the initial plasticity we observe in a naive ancestor. Are those plasticity being reinforced or opposed by subsequent evolutions? So to identify genes that show strong initial plasticity, we only using the grand ancestor, and we found those genes that show strong differentiation between 2 assay diet. And for the subsequent evolution, we are only use the constant cadmium and constant salt population. And then we use this model to identify genes that show strong differentiation in expression between constant cadmium population and salt population. And there are about 100 genes shared in these 2 categories. So if we plot the initial expression plasticity in the x-axis, so it is positive above 0, indicate the sample in the cadmium diet has higher expression than the samples in the salt diet for grand ancestor. For the y-axis, if it is above 0, indicates the cadmium evolved population has higher expression than the salt evolved population. And if the data fall into these two blue category, we will say they have a positive relationship that the initial plasticity is reinforced. But if it falls in these two categories, we think it's a negative relationship. Normally, we call this a counter-gradient response. And here's the data. And I think most of the genes show a counter-gradient response. And they have a pretty strong signals. So I think these patterns for gene counter-gradient for the gene expression plasticity seems emerging pattern. There are a couple other studies also found these patterns. For example, in guppies, I guess Professor Gallenborg may talk about this later today in guppies. So if we look at the data more carefully, but I won't show you here, the initial plasticity is mostly caused by many genes are overexpressed when the naive population encounter the cadmium diet. So the grand ancestor and the salt population never experiencing cadmium before, and they turn on their genes. And once they get adapted to the cadmium diet, the expression just reduces. So the exact mechanisms of this counter gradient is still unknown. It could be that the initial plasticity is bad, representing a perturbed expression, and then selection basically act on those expressions directly to bring back their optimum stage. But alternatively, the initial plasticity could be beneficial, at least transiently, because it's a stress response plasticity to help you to cope with cadmium stress. But once you adapt to cadmium, you no longer feel stress, you no longer to turn on those genes. So it's still kind of unknown in that case. But this plot is showing how counter gradient, how the evolved difference is reversing the plasticity, initial plasticity difference. Okay, so the first part of talk is mostly focused on when the population encounter a constant novel environment, how the effects of plasticity get opposed by the subsequent evolution. But how the adaptive plasticity evolved, and it actually required the environment to vary, and so how would heterogeneous environment shape plasticity adaptively? Well, as Maggie mentioned, selection could favor or disfavor plasticity. All the plasticity could be neutral, but it depends on what's the optimum trait value, optimum expression in these two environments. For example, here's some initial plasticity when the population encountered the environment. And if the optimum trait value is very different in these two environments, and once those heterogeneous population adapt to these two optimum, so selection bring them to move to the optimum, we will see an increase in plasticity. The similar logic apply that if the two optimum are very close to each other, and then selection basically could drive the expression or the trait value to the optimum, and eventually we will see a reduction in plasticity. And in terms of gene expression, we think across the whole genome, there should be some genes fall into select to be increased, and other genes select to be reduced plasticity. And how to identify genes in these two categories, we decide to use these two diet adaptive ancestor, so ancestral cadmium or ancestral salt. And the way we're doing is because we are regarding these ancestors, when they are put into their own diet, we think they are pretty well adapted to their diet. For example, for the AC, when they are assayed in the cadmium diet, these samples we think is pretty well adaptive. Then the expression of that sampled we think as an approximation as the optimum expression in that cadmium diet. Similar logic for the ancestral salt, when it's assayed in salt environment, their expression could be an approximation as the optimum expression. And to identify genes that show increased expect to equal to increased plasticity, we require the two optimum expression to be very different. And then initially, they show very little plasticity. So the AC is quite similar expression in both diets. And if the population experiencing both diets in history, they will move to the optimum and show increased expressions. And this increased expression plasticity. And this plasticity should have a directions. For example, in this case, the salt should have expression in the salt diet should have higher expression than in the cadmium diet. Then we could calculate the, this called adaptive plasticity, which is polarized by what's the direction in the two adaptive optimums. And basically this shown that, yeah, the two heterogeneity population show an increased evidence of increased plasticity compared to the two constant populations. And different groups just indicate they're significant different from each other. So this seems, okay, seems there's some, we could detect some signal of adaptive increased plasticity. So the same logic could apply for those we expect to select for reduced plasticity. And this time, just we've screened for genes with very similar optimum expression, but with high initial plasticity. So eventually, we expect those plasticity will gone if population adapt to both environment. Okay, but that's not the case. Only spatial seems to show a relative reduction in plasticity, but not the temporal. So this seems to suggest that the temporal heterogeneity seems not quite well adaptive to both environments. Is that the case? So to directly measure how to quantify how well they adapt to the environment, we just come up with a simple statistics called adaptive distance. And basically, we could treat what's the optimum expression as 0, and then we could treat the non-adaptive sample, their expression as 1. Then for each population, we could quantify them in between 0 and 1. So if that statistics is closer to 0, indicates it's close to the optimum expression. And we could do that in both diets. So first, for the set of genes that we expect to increase plasticity, this is expected. So the heterogeneity population are pretty close to the optimal, approaching to the constant adaptive population, and they are, and those non-adaptive constant population are way behind in the adaptive distance. So which is confirming the observed increased adaptive plasticity. However, for those genes that we expect to reduce plasticity, they still show the signal of adaptation. Both heterogeneity one are pretty close to optimum, and the temporal actually quite close and even maybe doing better than the spatial. So how can we reconcile basically the mean expression already close to optimal with observing seeming this maladaptive pattern for the temporal heterogeneity? Well, think there are some reasons we could come up with. So one possible explanation is that even though this optimum we set are very close to each other, and selection is supposed to drive the expression to the optimum, but if the adaptation is asymmetry, if it adapts much faster in one diet environment compared to on the other diet environment. So in that case, the plasticity may increase transiently during this adaptation until the adaptation finished on the other diet. So if that is true, then maybe suggests that the temporal heterogeneity would under a stronger constraint on selecting for reduction in plasticity. Or they have some stronger trade-off that the genes cannot approach to the optimum at the same time, resulting reduced in expression plasticity. I think this case seems to serve a reminder that selection is not truly acting on plasticity for many of the traits, but rather selection is acting on the mean expression in different diets and bring to the optimum, and then the plasticity kind of evolve indirectly because of that selection. That's the view of my word now. Okay. I think, yeah, we could summarize that we found some evidence of increased plasticity in the heterogeneous population, but not evidence of adaptive reduced plasticity. Although we found the adaptive evolution of expression for both sets of genes in both diets. And so the plasticity may evolve differently in temporal versus spatial heterogeneity. I think probably more theoretical work or empirical work comparing these two forms of heterogeneity, or even just the temporal heterogeneity when we try to vary in different conditions, like different frequency, may help us to understand how different forms of heterogeneity affect. And we also see strong patterns that those evolved difference in expression turn to counter those initial plasticity of their naive ancestors. So if this pattern is general, this may suggest us we may actually some work predict how gene expression evolve when the population encounter a novel environment if we could measure their initial plasticity. So how general this counter gradient patterns it is, right? So this setup is just in a simple laboratory condition with artificially chosen chemical stress. So I think next related project I'm working with John Poole in Wisconsin is just ask, does this counter gradient pattern still hold in natural population? So it's still ongoing. And John has still fruit fly population. They are originally from the warm environment in Africa. And then there are three separate expansion of those population expanded to cold environment. So in South Africa and Ethiopia, they expand to the local nearby highland. And for the Egypt, they expand to the France. And within each pairs of warm and cold population, the genetic distance are kind of short. But they already show a strong, significantly difference in cold tolerance between the population within each pairs. So right now we are just put those populations and rear them in either warm and cold environments to measure what's their expression plasticity. And we could also measure what's their adaptive change when they adapt to cold environment. So hopefully this project could just try to inform us a little bit more about the parallel counter-gradient patterns from this natural population with kind of independent selection event. So with that, I would like to thank all the people contribute to this work and this talk, and thank you for your attention.
So I think we have a little time for some questions. I might start us off, if you don't mind, April, and I'll pass. So Yuhang, I love this work. It's so exciting. And I think your sort of finding about the counter-gradient gene expression is really helpful in thinking about sort of where in a genotype-phenotype map we should, sort of what our expectation is for the evolution of plasticity based on where you are in the genotype-phenotype map. Mm-hmm. I wanted to ask you about something you said towards the end of your talk, which is about so selection operating on plasticity, on the phenotype itself rather than plasticity. And I think that might depend a little bit on your selection conditions. Yeah. So would you expect that to differ if you were sort of alternating flies within a single generation between those environments?
Yeah,
do you have any
a good
ideas?
point. Yeah. Like within generation variation, maybe plasticity is directly selected. Yeah. Right. I think that's a good point.
Thank you for your talk. I definitely enjoy reading your papers. And I have a small question that you have the ancestor of cadmium and ancestor of salt, and you made them to get your F1 generation. I wonder what's the performance of F1 in different conditions, and whether that plastic change and adaptive change are cis-regulated or trans-regulated, since it seems you could tell about that.
So the F1 plasticity.
And F1 adaptation, like whether the adaptive trait is dominant or the plastic trait is dominant due to trans factor. Thank you.
Yeah, I think definitely a great question. I don't know. Yeah, so we could always go back and then do the experiment. It seems, right, I would suggest that they could be both are quite adaptive to both diet. It seems the adaptation is a complex trait, and when we cross F1, at least we get whole genome from both adaptive population. But I don't know. Yeah, and potentially it's interesting to if it depends on what's the nature of selection, like what kind of genes they're acting on, and what's the dominance, and even epistasis. We may, at one time, I may think about those populations could separate, become some ecological speciation, so that may making the F1 doing pretty bad. But we haven't looked at them carefully. But thanks for the question.
So, great stuff. So I'm curious. You didn't talk much about the what these genes are, but like when you do the annotation, what are some of the interesting patterns that you're finding in terms of the identity of some of these genes?
Right. So in here we have 3 sets of genes. Like first set is analyzing counter gradient. We do some GO for all 3 sets. I think most of them did not show up any significant cluster pathway. For the first set, there's some transmembrane membrane stuff. So I definitely suspect some of them are related to stress response, but could also is just perturbate expression. I think both cases could be mixed in those genes. But yeah, we don't know. We don't have a significant, many significant geo categories. Yeah. Thank you.
Yeah, probably. Can you switch to the back? Yeah. Ah, you guys are set up. So do you need to hook this up to
Yes.
this thing here?
That thing. Yeah. Sorry. Let's see if that works. It worked earlier. It doesn't seem to be doing the thing where it.
Does that pull content too?
I don't think so. It's not sound. So. It's not connecting like it did.
Go ahead and hit view slideshow.
Okay. Yeah, definitely. Thank you. Yeah. There we go. If it's doing.
Oh, there's the mouse.
Maybe we need to go to the display thing again.
Yeah, I can get the mouse off that side here.
Sorry.
Go ahead, yeah.
Okay. Cool. Sorry about that. These things always work earlier in the day.
That's right.
Okay, so Karen Burkhardt is a postdoctoral fellow at the Smithsonian Environmental Research Center. One of the projects she's working on there is the role of local adaptation and phenotypic plasticity in freeze tolerance for 3 widespread mangrove species. Karen earned her PhD at Yale working with Oswald Schmitz. And we actually overlapped at Yale and shared some lab space for a while, so it's fun to have this chance to reconnect. Today, Karen will be sharing some of the results of her dissertation research, where she looked at how plasticity in plant defenses influences insect behavior, and then also alters the fluxes and flows of nutrients in old field communities. So thanks, Karen.
Hi. Sorry about that, everybody. So I'm very excited to speak here today. Thanks for the invitation. This is going to be a little bit of a change in pace from what we've been hearing about before, but I think Maggie may have set some of this up better, and I'll be able to move through quicker. This is a bit of an experiment. I'm trying to pull together a bunch of pieces of my dissertation at once, so you can let me know how it goes. Here we go. So anybody who's taken a general ecology course has probably come across a figure like this representing succession. So how we go from kind of disturbed, abandoned, old field habitats to forest environments. And I did my research in New England ecosystems. So you can't walk anywhere in Connecticut without coming across a stone wall in a forest. And initially, it sort of puzzled me, but what it turns out is that almost all of these habitats used to be abandoned agricultural fields in these systems. So when they put in their fields, they essentially put rock walls here, and so you see a signature of that on the landscape. And so one of the questions that motivated my dissertation work was how we go from these homogeneous tilled soils over here in early succession to, if you talk to an ecosystem ecologist, they will complain all day about how variable the soil conditions are in these forested ecosystems. And so one of the things that my lab works on is this kind of middle section of succession, and where these communities are really dominated by this one species, Solidago altissima, in our particular area. And so the question I had was whether kind of biotic factors could essentially change soil heterogeneity through traits of this dominant species. So yeah, over succession, how these biotic factors could alter soil heterogeneity patterns. And one way that people have thought about how species might impact ecosystem processes is through functional trait paradigms, where folks are looking at species mean trait values and then relating that to ecosystem processes. Now here, of course, I'm really talking about one species that's dominating the system, and so here we need more of an intraspecific approach. And I think the best way that this has been kind of thought about in the literature is through community genetics approaches, genes to ecosystems, and it essentially uses the quantitative genetic approaches that Maggie presented to try to link heritable genetic variation to ecosystem processes. One thing I want to point out here is that within these frameworks, we're often trying to minimize the environmental variation that we capture in these common garden type of experiments. And often we're collecting genotypes from over a wide range, geographical range. And so you might imagine that the genotypes we're collecting and doing experiments on represent a larger amount of genetic variation than we might see in a local field. And so one of the things I wanted to do was really focus on local scale, both environmental and genetic variation. And in order to do that, essentially, the typical sort of community genetics approach really focuses on genes, looks at heritability through genetic variation, and then links phenotypic variation to ecosystem processes. What I was interested in doing is really partitioning out, as Maggie alluded to, these different components of phenotypic variation, and then independently relating those to ecosystem processes. And so the basic way I'm going to go about doing this is give you an introduction to how we might think about herbivores impacting ecosystems in general. Then present a greenhouse experiment I did demonstrating plant defense changes in goldenrod genotypes across nutrient gradients. Then link that variation to legacy effects on ecosystem processes. And then lastly, touch on a multi-year field test that I did to essentially think about this whole framework within experimental raised bed system. So first, how do we think about how herbivores might affect ecosystems in general? So when my first year of grad school, I came across this paper by Mark Hunter. He's here in your department, so you're lucky. I noticed it I would like to think because of the great science in it, but also it had sentences like this. I've been thinking a lot lately about death and decomposition. I don't know if it's the onset of a midlife crisis or waiting for the academic deadwood in my own institution to be recycled to peak productivity. He wasn't at this institution at the time, so. But in any case, it kind of caught my imagination, and that's sort of what motivated the work that I'll be doing and presenting on today. And I would just like to highlight he has a new book out, essentially covering a lot of these concepts. You could have published it about 6 years ago when I was starting my dissertation. It would have saved me some time, but water under the bridge. Okay, so this is a simplified way we might think about how herbivores are going to affect ecosystem processes. So these blue lines are essentially direct effects of herbivores on ecosystem processes. So things like frass, carcass inputs, green fall, things like that. And we might consider these to be direct effects of herbivores on ecosystems. And in general, ecosystem ecologists usually discount these effects to a large degree because in general, in terrestrial systems at least, we only have about 10% of our overall biomass being consumed by herbivores. That varies, of course, but ecosystem folks usually don't pay too much attention to them. But of course, the other 90% of tissue that's going into these other pathways and pools has a signature of that herbivory, even if it's not being physically removed by the herbivores themselves. And so this can happen through changes in a bunch of traits, but also through root inputs, root exudates, that sort of thing. So people here, I don't have to spend too much time convincing that plants can respond plastically to herbivores. Some of the ways they can respond are through resistance responses that essentially deter herbivory. So these are some of the chemical defenses that Maggie was talking about earlier. There's kind of an alternative strategy that plants can take, though, as well. That's what we would think of as tolerance. And essentially, the plant might start regrowth responses or growth thinner leaves with the goal of essentially minimizing the negative impact of herbivores on their fitness traits. And these don't necessarily happen, they aren't mutually exclusive of each other, but they are kind of a way that we can think about how an intraspecific variation in traits might change ecosystem processes. So we can imagine if a plant is expressing tolerance traits, there's regrowth tissue, high quality on the landscape, we might see an increase in nutrient cycling as we have a higher quality and quantity of litter on the landscape. In contrast, resistance responses might result in lower quality or quantity of the overall tissue on the landscape and slow down cycling rates. And usually these things are thought of as either-or situations. They're not usually thought of in intraspecific frameworks. But what I'm going to argue is within a species, we can get kind of this multitude of responses. So how are we going to explore this? So first, I'm going to talk about specifically the plant defense strategies that this species, Solidago altissima, uses. And I think it's a really good species for thinking about these questions because it's a perennial clonal plant. So we can propagate it by cutting up pieces of rhizome material to get genetic clones, which makes it easier for these experimental structures. It's an obligate outcrosser and has these deciduous ramets that go up every year and then die back, but the rhizomes continue spreading out. And we know these species have a whole host of both induced and constitutive defenses that are a host of chemicals, structural responses, and tolerance responses. So they kind of run the gamut within the literature in these responses. Here you can just see the rhizome tissue that you can cut into pieces to grow clones. And I think this is an interesting system because essentially we can conceive of both genetic and herbivory-based mosaics. So when these species colonize an abandoned old field, there's about a 2-year period where seeds are coming into the system and are able to germinate and produce new plants. But then after that about 2-year recruitment period from seed, it's almost exclusively clonally based reproduction in the field. The clones are able to outcompete any new seedlings. So what you get is what you see here. These folks were great. They went out and dug up sections of old field and essentially genotyped all the individual ramets. So this is a 5-year-old field. So probably it's had about 3 years of clonal growth. And each of these colors is a genotype. And so within a meter, you can see that there's clumping of genotypes, but there's a wide diversity of genotypes on the landscape. They did the same thing with a 35-year-old field. And what you can see is many fewer genotypes, and what I would argue is probably happening is these are the edges of huge genotype clumps that are essentially taking up space on the landscape. And that's what I saw within my fields as well. So that field scale genetic mosaic, if there's genotype variation for these plant defense traits, could essentially result in trait differences that then alter decomposition patterns. So you get this mosaic of decomposition based on genetic identity. Alternately, you could also have a mosaic based on herbivore damage. So we also know that herbivore damage in these fields is very patchy. So these are kilometer fields down in Louisiana of goldenrod, just looking at herbivore damage. And it ranges from 0 to 60%. These are two different fields. Between fields, it ranges widely. So what you might imagine here is that if you have variable induction on the landscape, you could also end up with patchy trait expression, which could then lead to changes in the ecosystem processes in the soil beneath the plants. So the first thing I wanted to do was essentially quantify that genetic and environmental component to their plant defense strategies of these plants, and think about whether we can get at some of these E by E responses, which is something that the community genetics approach really doesn't get at very well because they want to minimize plasticity within a lot of these experiments. So I harvested 9 genotypes from one focal field. They're collected about 25 meters apart so that I know that I'm not getting the same clone, but there's no way they would have spread further than that within the 10-year period that this field had been growing. Grew them in the greenhouse across a nutrient gradient. So this is 4 levels of nutrient treatments of a full fertilizer. So it's not just nitrogen. I didn't want the plants to be limited in general by nutrients. And then basically, I impose treatments on clones of for constitutive or induced defense traits. And we'll get a little bit more into that. This is complex. I don't want you to understand it all. Basically, I could give a whole talk about the trait responses of these plants in general. What I want you to get out of this is essentially that genetic and environmental effects are complex on traits. So here on the x-axis, we have the nutrient treatment. And so here on the left side are plants that never have had an herbivore on them. This is leaf dry matter content. It's how much the plant is kind of allocating into the tissue of their leaves. And what you can see is here on the left side, leaf dry matter content really is an herbivory-driven trait. So when herbivores are present across the entire nutrient gradient, leaf dry matter content increases, regardless of genotype or the nutrient conditions. Here, chlorophyll, I would argue, is a little bit more of a nutrient-dominated trait because across this gradient, chlorophyll content increases regardless of herbivory. We also have genetically driven traits like leaf carbon content. This green genotype just has a completely different leaf carbon content than everybody else. And then lastly, we do have these E by E effects where across a nutrient gradient, leaf mass area decreases with increased nutrients unless herbivores are present, in which case you get no variation across the gradient. So this is just to say that these effects are very complex. I give a talk on it. But it's to make the argument that later on I'm going to use multivariate approaches to summarize kind of whole plant changes at once. And one of the reasons for that is that Maggie talked a lot about chemical defense. And that's an important part of defense responses and resistance in general. But it's actually kind of an integrated component of all of these things that can change in a plant at once. And so actually what I'm going to talk about, what I measured is almost everything here except specifically chemical defenses. So different perspective. Instead, what I did was essentially ask an herbivore. So this is Melanoplasma rubrum. This is the major dominant herbivore in these old field habitats that we work on. And essentially what I did was feed the plants to the herbivores and see how well they grew on plants of varying treatments. And here's what we find. So again, we've got nutrient treatment along the x-axis. Here we have resistance on the y-axis. And this is just a relative growth rate, an inverse of a relative growth rate. So what you can see is that resistance decreases across the nutrient gradient on constitutive plants. And this is kind of what we would expect. Plants are going to have higher nutrient content. Herbivores are going to grow better on them in the absence of nutrient limitation. Now, if you look at plants that have been previously fed on by herbivores, you see a completely different situation. So here, you essentially see a if anything, a slight increase in resistance across the nutrient gradient. And when we look at the genotype response ratios that we've got going on, what you can essentially see, this 0 line would be no induction at all. At low nutrients, there's induced susceptibility. So herbivores grow better on a plant if it's previously been fed on by herbivores, whereas at high nutrient levels, we have induced resistance here. So there's a flip-flop in kind of the strategies the plants are using where they only really induce a resistance response at high nutrient levels. So what happens with tolerance? So again here, sorry the axis got cut off, but this is nutrient level again. And so tolerance is that kind of ability for a plant to minimize the negative impacts of herbivory. And this is a little more complicated to measure than in Maggie's system because this is a perennial plant, so it produces asexually and sexually. I'm going to focus on asexual reproduction because I think it's one of the more important components for these plants spreading clonally in a field. But I'm happy to talk about other factors with folks. And what you can see is that this one line is essentially equal fitness between a damaged plant and an undamaged plant. So at low nutrient levels, we actually see plants are producing more rhizomes when they're damaged by herbivores than when they're undamaged. At high nutrients, there's a reduction. And I guess I've received the most pushback on this from any result I've had in my dissertation thus far. And I'm happy, it is also another talk. But I'm happy to talk about it with folks. I think what's happening is essentially what has been put forward by Wise and Abrahamson, the limiting resource model of tolerance, where at high nutrient levels, these plants are growing at maximum growth rate because they're not limited by nutrients. And then when herbivores feed on them, they're removing carbon and light gathering resources from the plant, so they can't recover from it. Whereas at low nutrients, that's not what's limiting the plant. Happy to talk about that more with other folks. But essentially, what we can see is that potentially this nutrient gradient could be driving these intraspecific variation changes. So what might that look like in terms of landscape level variability? So if we've got these nutrient levels on the x-axis again, and we've got palatability or decomposability and ecosystem process on the y-axis, you would predict that constitutive plants are going to steadily increase in palatability with increased resources. So if we have induced resistance at low phenotypes or low resource levels and tolerance at high, you would imagine that trait plasticity would essentially be increasing the variability across the landscape in your palatability or decomposability, whatever ecosystem processes you have. Now, the other option would be flip-flopped, right? Where you have a tolerance phenotype at low nutrients and induced resistance at high nutrients. And here, essentially, plasticity is decreasing variability in decomposition. And if you're paying attention, that's essentially what we're seeing in this system. We have tolerance at low nutrient levels paired with, if anything, induced susceptibility, and then we have resistance at high nutrient levels. So this is kind of the pattern we might see within this system. So to summarize this section, the same genotype can exhibit widely different strategies. And we're essentially going to be looking for this interaction, nutrient bioherbivory interaction, to see if there's a trait-mediated decrease in variation. And to do that, I essentially took the litter from this experiment and ran a decomposition experiment. So you place the litter in tubes, inoculate it with a common soil inoculum from the host field, and measure respiration of the microbes. So essentially, you measure the CO2 respired by the microbes and the litter mass loss. You can get a measure of decomposition efficiency. And so what did we find? So again, if the pattern holds and we're seeing similar relationships between palatability and decomposability, we would expect our treatments to equalize, induction to equalize variability. And that is indeed what we see. So here you've got decomposition efficiency on the y-axis. And if we compare that to our figure here, The solid line is essentially our expectation in the absence of herbivory, and the dotted line in the presence of herbivory. So it does appear that these trait changes that are occurring at the whole plant level on the landscape are translating to how microbes process litter when it enters the ecosystem. Okay, so how do we connect that in this way that we're interested in doing to ecosystem processes effects, partitioning out these components? And this is just a quick example I'm going to show you to kind of familiarize you. And what I'm doing is a redundancy analysis approach. And so essentially, it's a constrained PCA. So the axes I produce are only representing variation that was created by my experimental treatments. And so you build a trait space from leaf traits, and then post hoc, you fit a vector that represents decomposition in general. So building a trait space independently, seeing how it correlates with decomposition metrics. So this is what you can see when we look at only the genotype level effects. So I've removed herbivory and nutrient-based variation. And each of these colors is a different genotype. They're the 95% confidence intervals for that genotype. So you can see the green genotype is tougher than everybody else. That's kind of how you can interpret that. And we can explain a decent amount of the variation with genotype. Now, if we put on our decomposition vector, carbon mineralization strongly correlated with this genotype. And the R-squared values on this are like 0.4, something like that. Now, we can also do this by looking at the environment. So we remove the genotypic variation first. And so you can see all these circles are now centered. So basically, we've gotten rid of all the genotype variation. We're just looking at the environmental variation. And so nutrient treatments essentially increasing leaf in, herbivory treatments increasing leaf dry matter content, and a host of other traits. There's interaction happening. Post hoc, if we put on the decomposition frameworks, we can explain all of the metrics that I measured in the decomposition experiment. So basically, microbes are more efficiently decomposing litter that came from non-herbivory treatment plants at high nutrients. So this is one kind of approach to think about partitioning out that variance. So in summary, we kind of have both genetic and environmental components of this, and it's a result of many correlated trait changes. All right, so lastly, what I wanted to do is take these results and think about it in a landscape level framework. And so I put together a field experiment in order to do this. So we dug up an old field, built raised beds, filled them and homogenized them with 50% sand, 50% soil, split them in half. Half of them got nutrients, half of them didn't. Half of them got herbivores in a split plot design, half of them didn't. After the treatments, we added litter bags. The next May, I came back, sampled again, did soil samples, added treatments again, again did litter bags, and then the last summer we removed all the treatments. So basically we had 2 years of treatments and then a cessation. And again, we're using this little Melanoplasphema rubrum herbivore. There's going to be mesh here that you can see here separating the plants. So this is just a little slideshow intermission. This is kind of what happened for the plants. They came back up in the spring. They were caged. We harvested them. You can tell they reproduced quite prolifically over the course of the time. So 3 individuals went to 70 individuals often in the beds. So what's happening with trait expression across this time frame? So this is RDA again. Now we're actually having both genotype and herbivory and nutrients potentially in the model. Fortunately for us, before nutrients or herbivores were added, there's not a significant effect of them. It's only genotype driven. After we added nutrients, we can pick up a signature of that in the leaf traits on the landscape. After we add herbivores in 2013, we can pick up a signature of that on the leaf traits. By the second year of treatments, we can essentially pick up strong signals of both herbivory, nutrients, and genotype variation on the landscape. But then once we remove treatments, the variation associated with the environment goes away. So basically what I think is happening here is that we have these fast responses of plants to herbivory and nutrient addition, but then the genetic signature is more durable over the years. And what is that genetic signature? So these are 4 different genotypes. And they basically, essentially have different strategies. I guess I wasted my time early. So high litter quality, resistance here. And so we can think about that as contributing to this overall genetic mosaic landscape effect, because we have genetic components that are durable over time that are having effects, potentially below ground. And when we look at the processes below ground, we can detect signatures of genotype on the microbially available carbon content of the soil. So we are detecting some small alterations in ecosystem processes based on genotype. Now, if we partition out the environmental effects, we can explain a whole host of ecosystem processes. I don't have really time to go into all the things we measured, but essentially, a large number of things happening beneath the plant are related to the the environmental treatments. And so what I think if we can pull out from this is that spring microbially available carbon is higher under control plants that haven't had herbivores. That indicates that decomposition is faster in those treatments. But over the course of the season, we have a smaller decrease under herbivore plants under plants getting herbivory. And that indicates that we might have some carbon inputs from frass, exuvia, that sort of thing. All right, so just to sum up, oh, and you notice there isn't an herbivory by nutrient interaction here, and I'm happy to talk about that. Experiments don't always go how you planned. So in summary, I think what's happening is fast cycle carbon is increasing through these direct pathways, while we get a decrease in slow cycle carbon happening through these indirect plant-mediated pathways. So I think there is some evidence for ephemeral changes. So in summary, I think there is some evidence that the traits that are occurring on the landscape are playing a role in determining heterogeneity. We're getting at this a bit more through some gridded work that I won't go into. But I just will remind you that there's a lot of ecosystems out there that are dominated by a single species. And so we might want to think a little bit more about how intraspecific variation might be playing a role in these ecosystems. So with that, sorry for going over, and I'll take probably not questions.
Okay, thank you, Karen. We have coffee break just outside the doors here, and if you have questions for Karen, hopefully you'll ask them in the panel.
No, I mean, just taking 5 minutes at the beginning to get things set up never goes well.
That was really nice.
I wish yeah.
I love the experiment. That's really.
Yeah. Thank you. We'll see. I'm still trying to figure it all out. Yeah, no matter what you do, I feel like getting these things to work is tough.